Hello my dear students. In our last class, we have just started studying about the eukaryotic cells. We have discussed about cell membrane, the fluid mosaic model which was proposed by Singer and Nicholson, the functions of cell membrane, and I had also told you to practice the diagram of plant cell and animal cell. Students, please note, you have to show the presence of microvilli when you are asked to write about animal cell. Okay, please write microvilli when they ask you to write the animal cell diagram. Okay, yes. Let us proceed our study with the eukaryotic cell. Today, let us start our study with cell wall. Okay. Now, what is a cell wall? Cell wall is a non-living, rigid structure. Okay, non-living, rigid structure that is present outside the plasma membrane. I repeat. Cell wall is a non-living, rigid structure that is present outside the plasma membrane. Now, where do we find cell wall in eukaryotic cells? When it comes to eukaryotes, cell wall is seen only in plants and fungi. Okay, in eukaryotic cell wall is present only in plants and fungi. What is the chemical composition of cell wall, or what is the cell wall made up of? When it comes to algae, the cell wall is made up of cellulose, mannans, galactans and minerals like calcium carbonate. Okay, when it comes to cell wall of algae is composed of cellulose, mannans, galactans and minerals like calcium carbonate. In plants, the cell wall is made up of cellulose, hemicellulose, pectin and protein. Okay, when it comes to a plant cell, the plant cell wall is made up of cellulose, hemicellulose, pectin and protein. When the plant is young, the cell wall in a young plant is called as primary cell wall. Okay, when the plant is young, the cell wall that is present in a young plant cell is called as primary cell wall. This cell wall is capable of growth. Okay, this cell wall is capable of growth. It can increase its size. But later, okay, when the plant grows, okay, then this primary cell wall slowly diminishes. Okay, primary cell wall slowly diminishes and a new cell wall is formed, okay, inside the cell next to plasma membrane. If this is plasma membrane, here next to plasma membrane, here inside the cell. Okay, so earlier cell wall will be this, primary cell wall will be this. Now inside this, okay, inside this next to plasma membrane, new cell wall will be formed. Okay, now this cell wall is called as secondary cell wall, which does not have the ability to grow. Okay, secondary cell wall is formed in the later stages of life cycle of plants. Now, if this is the cell wall of one cell and if this is the cell wall of another cell, okay, like this, let us consider these two as cells. there is a structure which is called as middle lamella. Okay, there is a structure which is called as middle lamella. This middle lamella is made up of calcium pectate. Okay, this middle lamella is made up of calcium pectate which joins the two neighboring cells. Okay, so middle lamella is present between the two cell walls. Okay, cells which are adjacent to each other. The cell walls, okay, between the cell walls like this. Now this middle lamella, it acts as a glue. Okay, and it attaches it attaches the adjacent cell. The chemical composition of middle lamella is they are made up of calcium pectate. Okay. Let us study the functions of cell wall. When it comes to the functions of cell wall, the first important function is it gives shape to the cell. Okay, first function is cell wall provides shape to the cell. It provides shape to the cell. Cell wall helps in cell to cell interaction. Okay, it helps in cell to cell interaction. You know what is to interact with each other, right? Yes, so this cell wall helps in cell to cell interaction. Cell wall protects the cell from mechanical damage and infection. Okay, cell wall also helps in mechanical damage. That means if suddenly anything bombards with the cell, it will not the cell to break down very easily. It gives this mechanical support. Okay, so cell wall prevents the cell from mechanical damage and also infection. Okay, it prevents the entry of undesirable molecules. Okay, it prevents the entry of undesirable molecules. Okay, and then there is this middle lamella. Okay, there is this middle lamella. This middle lamella will help in the attachment of two adjacent cells. Okay, it helps to join the two adjacent cells like this. It acts as a glue and joins the two cells here. Fine. But remember students, in some places, if the cell is like this, okay, and this is middle lamella, in some places you can see that this is interrupted like this. You can see the presence of a structure like this which transverse the middle lamella and the cell wall. Okay, transverse ಮಾಡೋದು ಎರಡನ್ನೂ ಕಟ್ ಮಾಡಿದೆ ಇಲ್ಲಿ, cell wall as well as middle lamella. Okay, both are transversed here. Now this structure is called as plasmodesmata. Okay, this structure is called as plasmodesmata. This connects the cytoplasm of one cell with the cytoplasm of other cell. Okay, this plasmodesmata which transverse the middle lamella and the cell wall. Okay, this helps in the connection of cytoplasm of one cell with the cytoplasm of the adjacent cell. Okay, so that is about the cell wall. The function of cell wall is it gives shape to the cell. Okay, it provides definite shape to the cell. So it helps in cell to cell interaction. It gives mechanical, it prevents the cell from mechanical damage and infection. It prevents the entry of, the cell wall prevents the entry of undesirable molecules. And then middle lamella, the layer which is made up of calcium pectate, which is present between the two cell walls, okay. This helps in the joining, okay. It acts as a glue and it joins the two cells, okay. It helps in joining of the two cells. The middle lamella and the cell wall is transversed, okay, by a structure called as plasmodesmata. This helps in the connection of the cytoplasm of one cell with the cytoplasm of the next cell, adjacent cells, okay. Cytoplasm to cytoplasm connection is formed with the help of this plasmodesmata. That is about cell wall. Next, let us study about endoplasmic reticulum and then Golgi apparatus, lysosomes and vacuoles, okay. Students, we all know that these are all cell organelles, right? We all know that endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles are cell organelles, but since all of their function coordinate with each other, okay, all they work together, their functions coordinate with each other, together they form endomembrane system, okay, together they form endomembrane system. I hope you understood. Endoplasmic reticulum, vacuole, lysosomes, Golgi apparatus, idu nalku kuda they are cell organelle, okay. But all these cell organelles, idra elladra functions coordinate, dependent on each other, they coordinate, jotege kelsa madodrinda ee nalku organelle seri adanna navu ee nalkanna matra we call it as endomembrane system. In that, let us first study about endoplasmic reticulum, okay. Endoplasmic reticulum. Endoplasmic reticulum. Now, if this is a cell and if this is a nucleus, endoplasmic reticulum is found as a reticulum or network, okay. These are actually tube-like cells and you find these cells near the nucleus like this, okay. So endoplasmic reticulum are reticulum or tube network of, okay. It is reticulum is nothing but network, okay. Ondakkondu join agiratte anta, okay. They are the reticulum or network of tube-like structures that are present near nucleus, okay. This endoplasmic reticulum, okay, they divide the cytoplasm into two compartments, okay. This endoplasmic reticulum divide the cytoplasm into two compartments like this. So one which is present inside endoplasmic reticulum, if this is a tube-like structure, okay, if this is a tube-like structure, this surface which is present within the tube, that is called as luminal surface. Artha agtide alva? Ee tara tube-like structure present agidre, okay, the surface that is present inside here, this is called as the luminal surface, and this surface which is exposed to cytoplasm, okay, the surface which is exposed to cytoplasm, it is called as extraluminal surface, okay, extraluminal surface. I hope you understood, okay. Endoplasmic reticulum hege iratte? It is a network of tube-like structures, network ondakkondu attach agi iruvantadu, network of tube-like structures. Yelliratte, that is present near the nucleus. Okay, this endoplasmic reticulum divides, okay, the cytoplasm into compartments. Okay, hege. So if this is endoplasmic reticulum, ee tube-like structure na olagade iruva jaga, that is luminal surface. Okay, the surface that is exposed to cytoplasm is called extraluminal surface. Okay, yes. The endoplasmic reticulum, sometimes this endoplasmic reticulum, okay, that is attached to ribosomes. It is attached to ribosomes. If the endoplasmic reticulum is attached to ribosomes, or if you can see the presence of ribosomes on an endoplasmic reticulum, then you call such endoplasmic reticulum as rough endoplasmic reticulum. Okay, if you see ribosomes attached to endoplasmic reticulum, then you call them as rough endoplasmic reticulum. Okay, if the endoplasmic reticulum does not have the presence of ribosomes on them, then they are called smooth endoplasmic reticulum. This is an important question for 2 marks. Okay, differentiate between rough endoplasmic reticulum and smooth endoplasmic reticulum. If endoplasmic reticulum has ribosomes on its surface, then it is called as rough endoplasmic reticulum. If endoplasmic reticulum does not have ribosomes, ribosomes are not attached to its surface, then it is called smooth endoplasmic reticulum. Okay, coming to the functions of endoplasmic reticulum, since ribosomes are attached to rough endoplasmic reticulum, I told you in the last class, the main function of ribosomes is they are involved in the synthesis of proteins. So, rough endoplasmic also performs the function of protein synthesis. Okay, the function that is performed by endoplasmic reticulum is rough endoplasmic reticulum. Okay, it is involved in protein synthesis. Okay, protein, the production of proteins, they are involved in protein synthesis. When it comes to smooth endoplasmic reticulum, they are involved in the production or synthesis of lipids. Okay, smooth endoplasmic reticulum produces lipids, and this lipids, the production of lipids is especially seen in animals. Okay, that is the function of endoplasmic reticulum, production of proteins by rough endoplasmic reticulum and production of lipids by smooth endoplasmic reticulum. This is the first organelle of endomembrane system. Okay, let us study about the second organelle of endomembrane system, that is Golgi apparatus. Okay, the second organelle in the endomembrane system is Golgi apparatus. This Golgi apparatus was discovered for the first time by a scientist called as Camillo Golgi. Okay, this Golgi apparatus was discovered by the scientist called as Camillo Golgi, hence it is named after him as Golgi apparatus. Okay, these Golgi apparatus are flat. Okay, they are flat structures. They are disc shaped sacs. Okay, they are disc shaped. You know how a disc will be present. Disc shaped sac andre cheela anta gottide, bag. Okay, disc shaped sacs. Athwa we can also call them as cisternae. Okay, we can also call them as cisternae. This structure can also be called as cistern. So when it comes to Golgi apparatus, it is flat disc-shaped sacs, or we can also call them as cisternae. Usually, this Golgi apparatus or the cisternae will be stacked parallel to each other. Okay, they will be stacked parallel to each other. Stacked ಅಂದ್ರೆ ನೀವು ಬುಕ್ಸ್ ಅಥವಾ ಕಾಯಿನ್ಸ್ ನ ಹೇಗೆ ಒಂದರ ಮೇಲೆ ಇಡ್ತೀರಲ್ವಾ, ಅದನ್ನ ಸ್ಟ್ಯಾಕ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಬುಕ್ಸ್ ಅಥವಾ ಕಾಯಿನ್ಸ್ ನ ಇಡುವ ರೀತಿ ಹೀಗೆ ಒಂದರ ಮೇಲೆ ಒಂದಿರ್ತಾರಲ್ಲ, ದಟ್ ಇಸ್ ಸ್ಟ್ಯಾಕ್ಡ್ ಅಂತ, ಆ ಥರ ಇಡೋದನ್ನ ಸ್ಟ್ಯಾಕ್ಡ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ದೇ ಆರ್ ಸ್ಟ್ಯಾಕ್ಡ್ ಪ್ಯಾರಲಲ್ ಟು ಈಚ್ ಅದರ್. ಓಕೆ, ಸ್ಟ್ಯಾಕ್ಡ್ ಪ್ಯಾರಲಲ್ ಟು ಈಚ್ ಅದರ್. ಅಂಡ್ ದೆನ್ ವೇರ್ ಡು ಯು ಫೈಂಡ್ ದಿಸ್ ಗಾಲ್ಗಿ ಅಪ್ಯಾರಟಸ್ ಅಂತ ಕೇಳಿದ್ರೆ, ಯೂಶುವಲಿ ಇಟ್ ವಿಲ್ ಬಿ ಪ್ರೆಸೆಂಟ್ ನಿಯರ್ ದ nucleus. Okay, usually they will be present near the nucleus. So near the nucleus, if this is the nucleus, you can see Golgi apparatus here. So nearby, first it will be like this. Later it becomes like this. Okay, so near to the nucleus, the cisternae will be convex. Okay, it will be convex. Here it is called as cis phase, or now it is called, or you can also call them as forming phase. Okay, so near the nucleus, the Golgi apparatus will be convex shaped. Okay, it will be convex shaped. Now this is called as, it is in its cis phase or forming phase. A little bit away from the nucleus, you can see the Golgi apparatus will be concave. Now it is called its trans phase or maturing phase. Okay, trans phase or maturing phase. Fine, I repeat. Where do you find the Golgi apparatus? Near the nucleus. But near the nucleus, here, very near to the nucleus, it will be in convex shape. If it is in convex shape, you call it as cis phase or forming phase. A little bit away from the nucleus, you can see the cisternae. It will be in concave shape. Now you will call it as, it is in its trans phase or maturing phase. Definitely the two phases are different. Okay, they are different, but both will be interconnected to each other. Okay, the phases are different. They have different functions, but both are interconnected. Okay, let us see the function of this Golgi apparatus. When it comes to the function of Golgi apparatus, the first one is, the main function of Golgi apparatus is, it helps in packaging the material. Okay, it helps in packaging. Okay, packaging material. Whatever needs to be sent to other organelles. Okay, Golgi apparatus will pack it and send it to the target organelles. Okay, so packaging, Golgi apparatus is the packaging material. Second thing is, it is the site of production of glycoprotein and glycolipid. Okay, it is the site of production of glycoprotein and glycolipid. Next. Okay, so whatever materials are received from endoplasmic reticulum, okay, whatever materials are received from endoplasmic reticulum, okay, that is packed here. Okay, that is packed here. Here the cis, okay, this cis phase will receive that material in the form of vesicles. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಯಾವುದಾದ್ರೂ ಒಂದು ವಸ್ತು ಈಗ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿದೆ, ಪ್ರೊಡ್ಯೂಸ್ ಆದಮೇಲೆ ಇಟ್ ವಿಲ್ ರೀಚ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ವೆಸಿಕಲ್ಸ್. ಈ ಥರ ಗುಳ್ಳೆಗಳ ಹತ್ರ ವೆಸಿಕಲ್ಸ್ near the cis form. Okay. After this, slowly now, it will move like this. Okay, it will move like this, and later, okay, when it becomes concave, illi maturing phase alli it gets packed. By the time it comes to hige barta hogi, by the time it comes to maturing phase, it will get packed, and from there it will be sent to the target organelles. Okay, na yellige hogbeku within the cell athwa outside the cell allige ಕಳಿಸುತ್ತೆ. Okay, so the materials that is received from endoplasmic reticulum, okay, materials received from endoplasmic reticulum that reaches the cis phase, in the form of vesicles, okay, in the form of vesicles, and then it will be packed. Okay, then the maturing phase will send it to the target organelles. Okay, the next one is the ribosomes that is present on endoplasmic reticulum. Okay, that is present in the endoplasmic reticulums. They produce proteins. The packaging of proteins is also done by Golgi apparatus. Okay, the proteins that are produced by ribosomes, which are attached to endoplasmic reticulum. Okay, the proteins produced by ribosomes attached to endoplasmic reticulum, those proteins are also packed by Golgi apparatus. So this is the function of Golgi apparatus. I repeat, Golgi apparatus was discovered by scientists called as Camillo Golgi. Okay, it is a densely stained structure present near nucleus. They are disc shaped, tube like. Okay, athava you can just call them, they are cisternae. These cisternaes near the nucleus, they will be in the form of concave, or ಆ ಫೇಸನ್ನ ಸಿಸ್ ಫೇಸ್ ಅಥವಾ ಫಾರ್ಮಿಂಗ್ ಫೇಸ್ ಅಂತ ಕರಿತೇವೆ. ನಿಧಾನಕ್ಕೆ ಸ್ವಲ್ಪ ಅವೇ ಹೋದಾಗ ಕಾನ್ವೆಕ್ಸ್ ಶೇಪ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಇದನ್ನ ಟ್ರಾನ್ಸ್ ಫೇಸ್ ಆರ್ ಮೆಚೂರಿಂಗ್ ಫೇಸ್ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಇದೆರಡರದ್ದು ಫಂಕ್ಷನ್, ಸಿಸ್ ಫೇಸ್ ಅಂಡ್ ಟ್ರಾನ್ಸ್ ಫೇಸ್ ಎರಡರದ್ದು. ಓಕೆ, ಅಂದರೆ ಫಾರ್ಮಿಂಗ್ ಫೇಸ್ ಮತ್ತು ಮೆಚೂರಿಂಗ್ ಫೇಸ್ ಎರಡರದ್ದು ಫಂಕ್ಷನ್ಸ್ ಡಿಫ್ರೆಂಟ್, ಬಟ್ ದೇ ಆರ್ ಇಂಟರ್ಕನೆಕ್ಟೆಡ್. When it comes to functions of Golgi apparatus, first thing is they are very good packing materials. Golgi apparatus is the site where the glycoprotein and glycolipids are formed. The materials that are produced by endoplasmic reticulum are packed in Golgi apparatus. Okay, before packing, what happens? The product reaches the cis phase in the form of vesicles. After packing, then okay, the maturing phase sends this product to the target organelles. Ribosomes produce proteins. Okay, these ribosomes are attached to endoplasmic reticulum. So the proteins produced by them are also packed and transported by Golgi apparatus. That is the second organelle of endomembrane system. Now, the third organelle of endomembrane system is lysosomes. These are membrane bound organelles that are produced during the process of Golgi apparatus. Okay, this is a membrane bound structure. This is a membrane bound structure. Yaru produce ಮಾಡ್ತಾರೆ? By the process of Golgi apparatus. This lysosome, okay, it is very rich in hydrolytic enzymes. Okay, they are very rich in hydrolytic enzymes. So enzymes like lipase, protease, carbohydrase. Okay, these enzymes are present within the lysosomes. Okay, hydrolytic enzymes like lipase, protease and carbohydrase are present inside lysosomes, but all these enzymes are active only at acidic pH. Okay, all these are active only at acidic pH. Now, when it comes to functions of lysosomes, we have to know that the main function of lysosome is it will help in the digestion of lipids, proteins, and carbohydrates, along with this also nucleic acids. Okay, all these are digested ಅಥವಾ broken down. We know that enzyme in a ಕೆಲಸನೇ break down ಮಾಡೋದು. Okay, you will study in detail about the enzyme in your next chapter. Okay, next chapter ಅಲ್ಲಿ enzymes ಅನ್ನ ಡೀಟೇಲ್ ಆಗಿ ಓದ್ತೀರಾ. ಈಗ ಇಷ್ಟೇ ಸಾಕು. ಏನು ಅಂದರೆ ಸೊ ಲೈಸೋಸೈಮ್ ಕಂಟೈನ್ಸ್ ಹೈಡ್ರೋಲೈಟಿಕ್ ಎನ್ಸೈಮ್. ಇದರೊಳಗಡೆ ಹೈಡ್ರೋಲೈಟಿಕ್ ಎನ್ಸೈಮ್ಸ್ ಇರುತ್ತೆ ಲೈಕ್ ಲೈಪೇಸ್, ಪ್ರೋಟಿಯೇಸ್, ಕಾರ್ಬೋಹೈಡ್ರೇಸಸ್. ಇದು ಯಾವಾಗ ಮಾತ್ರ ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ ಅಂದರೆ ಓನ್ಲಿ ಇನ್ ಅಸಿಡಿಕ್ ಪಿ ಹೆಚ್. ಓಕೆ, ಯಾವುದ್ರ ಮೇಲೆ ಆ್ಯಕ್ಟ್ ಮಾಡುತ್ತೆ ಅಂದರೆ ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಇದ್ರ ಲೈಸೋಸೋಮ್ಸ್ನ ಕೆಲಸನೇ ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಬ್ರೇಕಿಂಗ್ ಡೌನ್ ಆರ್ ಡೈಜೆಸ್ಟಿಂಗ್ ಲಿಪಿಡ್ಸ್, proteins, carbohydrates and nucleic acids. Okay, yes. Next we will study about the last endomembrane organelle. Okay, that is fourth endomembrane organelle, vacuole. Okay, we'll study about vacuole. Vacuole is a membrane bound space. Okay, it is a membrane bound space that is present in the cytoplasm of both plant and animal cell. In plant you can see like this, a large vacuole, whereas when it comes to animal it will be a bit small. Okay, so vacuole is a membrane bound space that is found in the cytoplasm of plant and animal cell. This vacuole has a single membrane which is called as tonoplast. Students, this is very, very important. Usually asked for 1 mark. Name the membrane of vacuole. Okay, the membrane of vacuole is called as tonoplast. Now, what is present within this vacuole? We can see the presence of water, the presence of sap. What is sap? Water along with the dissolved organic salts. Okay, water along with dissolved organic salts are called as sap. And then there are also excretory products whichever is not needed by the plants. ಗಿಡಕ್ಕೆ ಯಾವುದು ಬೇಡ. Okay, so vacuole consists of water, it consists of sap, it consists of excretory products that are not required by the plants. Okay, when it comes to vacuoles, this membrane is very important because the ions that are present in the cytoplasm is transported into the vacuole against the concentration gradient. I told you in the previous class, concentration andre gottu namge, gradient andre difference. Okay, gradient andre difference anta. So ಸಾಮಾನ್ಯವಾಗಿ higher concentration ಇಂದ lower concentration ಗೆ movement ಆಗುತ್ತೆ ions. ಹೌದು ತಾನೆ, higher ಇಂದ lower ಗೆ ಆಗಬೇಕು. But in plants ಅಯಾನ್ಸ್ನ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಲ್ರೆಡಿ ಇಲ್ಲೇ ಜಾಸ್ತಿ ಇದೆ ಒಳಗಡೆ. ವ್ಯಾಕ್ಯೂಲ್ನ ಒಳಗಡೆ ಆಲ್ರೆಡಿ ಅಯಾನ್ಸ್ ಜಾಸ್ತಿ ಇದೆ. ಇನ್ನೂ ಕೂಡ ಇಲ್ಲಿ ಹೊರಗಡೆ ಇರುವಂಥ ಅಯಾನ್ಸ್ನ ದಿಸ್ ಮೆಂಬ್ರೇನ್ ಗೆಟ್ಸ್ ಇಟ್ ಇನ್ ಟು ದ ವ್ಯಾಕ್ಯೂಲ್. ಅಂದರೆ ಎಗೇನ್ಸ್ಟ್ ದ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಗ್ರೇಡಿಯಂಟ್ ವ್ಯಾಕ್ಯೂಲ್ನ ಒಳಗಡೆ ಅಯಾನ್ಸ್ ಬರ್ತಾ ಇದೆ. ಓಕೆ, ವ್ಯಾಕ್ಯೂಲ್ನ ಒಳಗಡೆ ದೇರ್ ಇಸ್ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ಅಯಾನ್ಸ್. That is why the membrane here is very important, the tonoplast. Okay, when it comes to the function of vacuole, we have to remember 2 important functions. The first function is in amoeba we can see the presence of contractile vacuole. Kelidira alva, Amoebadalli there is presence of this contractile vacuole. The function here is it helps in excretion. Okay, it helps in the process of excretion. When it comes to when it comes to protesta, we can see the presence of food vacuole. This food vacuole is very helpful in engulfing the food, capturing the food. Okay, engulfing the food. So if they ask you the functions of vacuole, you will write two functions. The first function is in amoeba, contractile vacuole helps in excretion. In protesta, the food vacuole helps in engulfing, andre capture madodu anta, engulfing the food. Okay, I'll repeat again. Vacuole, the membrane bound structure found in the cytoplasm of plant cell and animal cell. Okay, so this membrane in a vacuole is called as tonoplast. That is very, very important. Within the vacuole we can see the presence of water, sap, and then excretory products whichever is not required by the plants. Okay, the membrane of vacuole is very important because it helps in the movement of ions against the concentration gradient. Fine. Okay, so this is about the endomembrane system. Endomembrane system anta yaake helteve alli? 4 organelles, their functions are coordinated. Okay, so the organelles under endomembrane system are endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. Please go through the NCERT, practice all these once again. Okay, let us meet in the next class. Thank you.